Hi, this is Ms. Delosier, and this is questions 1 through 10 of our long metabolism review. So let's go ahead and get started on this. Which of the following is most similar in structure to ATP? ATP stands for adenosine triphosphate. That's an adenine, a ribose sugar, and a phosphate. And the adenine and the ribose together make adenosine, and a pentose sugar can be ribose. A nucleotide, a DNA nucleotide, does in fact can have an A in it, can have an adenine. RNA molecule definitely has a ribose and can have adenine in it, and it also is going to have phosphates because the phosphates and the sugars actually make up the backbone of a nucleic acid. An amino acid, no, and a phosphate group, yes, and a phospholipid. I got the phosphates. So what's going to have the most in common? That's going to be C. Which of the following statements is true about enzyme-catalyzed reactions? Well, an enzyme-catalyzed reaction is going to go ahead and lower the activation energy. So it lowers the amount of energy that has to be added to a reaction, but you're going to get the same end product out later. So it lowers the initial cost, but it does go ahead and speed up a reaction. So my answer is going to be A. The reaction rate is faster than the same reaction in the absence of an enzyme. The only other one here that really makes sense is you might think that an enzyme catalyzed reaction releases more free energy than non-catalyzed, but you got to remember on this the curve, you're just lowering the hill, but this is your releasing of your free energy, and the free energy is the same. What's different is how much energy you put in. So if it's a lower hill, you're going to get up it faster. So my answer there is A. So that is that. And let's clear that and scroll down. So what is the term for metabolic pathways that release stored energy by breaking down? Breaking down is my key word here. Complex molecules. When I'm building stuff up, like if I'm getting stronger, taking steroids, that's anabolic steroids. We want the opposite of my anabolic, so that's going to be catabolic. So that's going to be answer B, and we don't really need to talk about the rest of those. Which of the following statements describes the results of this reaction? So let's look at the reaction. C6H12O6 plus 6O2 yields 6 carbon dioxide plus 6 water. So that is sugar plus oxygen is going to yield carbon dioxide plus water plus energy. So this is cellular respiration, and it's balanced. So what am I doing? I'm adding sugar to oxygen. Anytime you add anything to oxygen, you are oxidizing that thing. So if I let an apple sit out in the air, I'm oxidizing the apple, and that's why it turns brown. So when I expose the glucose to oxygen in your mitochondria, what you're doing is you are oxidizing your sugar. So we don't want this one because that says that I am reducing my sugar. So that one is clearly false. Now, if I'm oxidizing the sugar, then the opposite is that something else has to be reduced. So my sugar is oxidized and my oxygen is reduced. That sounds mostly right, so let's look. Am I adding oxygen to my oxygen? No, so I'm not oxidizing my oxygen. That's weird. Carbon dioxide is reduced and oxygen is oxidized. We just said that that's not happening. And am I adding oxygen? Here it says I'm adding oxygen to my carbon dioxide, and I'm not doing that. So my answer here is A. When a molecule of NAD+, this is cellular respiration, there's no P, right? So when a molecule of NAD+ gains a hydrogen atom, so I'm going from NAD+ to NADPH, I add a hydrogen. Then the molecule is add an oxygen, that's oxidized, add a hydrogen, that is reduced. So I added a hydrogen, so I am reducing it. That's pretty clear cut. We don't even have to work through that one. So let's go ahead and scroll down to the next page and work through 6 through 9 real quick. Where does glycolysis take place in eukaryotic cells? Glycolysis takes place in the cytosol. The oxygen consumed during cellular respiration is involved directly in which process or event? Oxygen is involved in the electron transport chain. That's electron to the next one. So the oxygen is at the end of the electron transport chain, and you remember its role was to accept the electrons at the end of the electron transport chain. Just so you know, the citric acid cycle is the Krebs cycle. It's the same thing. You don't oxidize pyruvate. There's no oxygen with the pyruvate, right? So this is B. Which process in eukaryotic cells will normally proceed whether oxygen is present or absent? Well, that's going to be glycolysis, because glycolysis happens outside of the mitochondria. There's no oxygen required for that. The other thing that you could have answered there would have been fermentation. You could have had fermentation, but that's not an answer. In addition to ATP, what are the end products of glycolysis? So when you do glycolysis, you add a glucose, okay, and then you get out pyruvate, and specifically you get out 2-pyruvate. Now, what else do you get out? You put in, and I'm going to generalize this, 4 ATP. Actually, I'm sorry, that is incorrect. You put in 2 ATP, and you get out 4 ATP. So your net there is 2 ATP. And then you're also going to get out 2 NADPH. So what am I making at the end of glycolysis? I'm making pyruvate, NADPH, and ATP. I'm not making carbon dioxide. I'm not making carbon dioxide. I'm not making carbon dioxide. I'm not making FADH. So I'm making NADH and pyruvate. That's my answer. And the last one that we're going to do on this video is number 10. Number 10, the transport of pyruvate into mitochondria depends on the proton motive force. This translates into the hydrogen gradient. That's what that means. Across the inner mitochondrial membrane. So you have to have a proton gradient. So how does pyruvate enter the mitochondrion? And it enters the mitochondrion through active transport. So you have to get the pyruvate, which is, I mean, pyruvate's a 3-carbon sugar. So it's a 3-carbon sugar, right? So think about it, just what you know about the cell membrane. I have a 3-carbon sugar, and I have to get it inside the mitochondria through a phospholipid bilayer. So how do you get anything that's that big in through a phospholipid bilayer? Through a protein channel. So it's not just going to go on its own. So you're going to either have to do facilitated diffusion or active transport. This says force, that implies energy. So let's go with active transport. So I will continue with question number 11 in the next video.